The project we've been working on will be used in various fields of biomedical and biological engineering, such as DNA research and tissue engineering. This device is made to create a constant magnetic field that cells can be placed in and examined under a microscope, assisting researchers by giving them ease of access to examine this effect with their device will further advance developments in the biomedical or biological field. Our advisor, Dr. Marathamathu, will be utilizing her device in his cellular mechanical biology lab.
We will now go over the methods for designing a 3D prototype. The requirements of the device for our advisor's research were that it had to be compatible with Leica DMI-8 fluorescence inverted microscope, be biocompatible, and lastly have the ability to apply or remove magnetic force without disrupting the specimen. This is our finalized chamber design, which was drawn using Fusion 360. The bottom piece is the chamber for holding cells, magnetic beads, and nutritive substance on a glass cover slip, and the top pieces are the interchangeable magnet housing rings.
To accommodate the requirement of being able to remove the magnetic field, we attach wooden dowels to the magnet housing rings. This is important as experiments will be conducted on a microscopic scale, and these will keep the specimens from being jostled. This slide shows the bottom view of the magnet housing. We designed two different interchangeable magnet housings. The larger ring holds double the magnets. The magnets in the rings are arranged in a Halbach array. We chose to use N52 neodymium magnets as they are the strongest on the market. These magnets will exert a force on the beads on the order of magnitude of 10 nanonewtons. The Halbach array augments the magnetic field so that it is maximized toward the side of the device where the cells on the cover slip will be located, while canceling the field to nearly zero on the other side. Also maximizes the magnetic forces along the XY plane while minimizing those in the Z direction.
The device was then able to be 3D printed and picked up from the lab. Here is a picture of the printed device. The material that was used to 3D print the device was Vera White Plus, which meets the biocompatibility requirement. Also, since the size restraints for the material are so small, the Vera White gave a strong bond and a smooth finish. Next, the magnets were installed into place. The arrows denote the dipole moment vectors which go from south to north. After the pole locations were found, the magnets were superglued to the bottom of the device.
This device will be used with a cover slip layered with soft silicone, fluorescent beads, and a magnetic bead. The magnetic bead will be attached to the soft silicone with connecting proteins, FC protein, and protein A. This cover slip assembly will be the reference image. When the cover slip goes into the device, the magnetic bead will be pulled toward the perimeter, pulling along nearby fluorescent beads. Using PIV software, a displacement map can be created along with a force stress map showing the stress generated on the surface of the silicone. It's expected that only the fluorescent beads in close proximity will be affected and that other beads will not have an effect. As you can see in this video, you can see a small displacement of the fluorescent bead. Because of this movement, our device is functional.
We would like to give a big thank you to Dr. Venkat Marathamathu for being such an amazing advisor, as well as a shout out to the ODU Modeling and Simulation Lab for printing our device.